Oh, I muted. Okay. Hi, everyone. Welcome. Thank you for coming. So my name is Ariel Parker, and I'm the Assistant Director of Education and Diversity for CEMD. And this summer, I've had the pleasure of helping to facilitate our undergraduates expanding boundaries summer research program here at UPenn. We appreciate all of you for coming in person or on Zoom to come learn about the research that CEMD UExB students have done this summer. To the summer researchers, we hope you have enjoyed working in CEMD labs and our programming within the center. We also hope you had a great summer in Philly and at Penn. We look forward to seeing most, if not all of you at BMES this year in Baltimore. So we hope that today is more of a see you later than a goodbye. Beyond BMES, we encourage you to stay in touch with us about your accomplishments and your career progressions. Special thanks go to Dr. Amanda Catone, Dr. Annie Jones, and Dr. Avery Williams-Jones for their work in planning and coordinating the logistics for this program. Thank you to your mentors for all their involvement with you and their work behind the scenes and learning how to be great mentors. Thanks also to the principal investigators for hosting you in their labs. And finally, thanks to you, summer researchers. We are proud of you and the work you've done. With that, let's start with our first speaker. Should introduce you. I'm a horrible host. I should introduce him. So everyone, our first speaker is going to be Mano Surti from University of Connecticut. This summer he was in the Berkeley lab, and today he's going to tell us, shouts out, and today he's going to tell us about his project investigating YAP-TAZ dynamics in tendon-tensile homeostasis, a two-pronged approach. So with that.
Good morning, everyone. Thank you all for joining. Today, I'm going to be talking about the research that I conducted over the past 10 weeks with Elizabeth Seidel and Dr. Joel Burkle on investigating the dynamics of YAP and TAZ in tendon-tensile homeostasis. So first, a little bit of background on tendons. Essentially, they function to transmit forces from muscle to bone by undergoing load cycles. So they always maintain some sort of residual tension. Loss in this tension can result in imbalances with extracellular matrix turnover, which is specifically shown by increases in matrix metalloproteinases, or MMPs. These are proteins that are catabolic to the ECM. The mechanics that mediate these early responses are pretty unknown. By studying the mechanotransductive response of the tendon fibroblasts that we're studying, we can better understand the onset of tensional homeostasis loss. So to do that, we're going to be zooming in on YAP and TAZ, which are transcriptional regulator proteins that are activated by growth factors, signaling pathways, and mechanical triggers. So this basically means that they're proteins that bind to different DNA sequences to regulate the expression of certain genes by up or downregulating their transcription. Now a little bit about their mechanistic behavior. So YAP and TAZ form a complex with TEAD in order to translocate into the nucleus and affect genetic outcomes. So their activity is basically controlled by their cellular location, whether they're in or out of the nucleus. One principal trigger of them is the cytoskeletal forces of the cells and also the stiffness of the extracellular matrix that they exist in. They're very intriguing in orthopedic tissues and have recently been implicated in a study of tendon conditioning. So this previous investigation shows that a depletion of YAP and TAZ shows similar gene expression to a loss of tension. And during this loss of tension phenotype, we see increases in these MMPs. So to further explain this, here we have this tendon fibroblast. It's among its really abundant extracellular matrix. It's really gripping onto those proteins in a state of happy homeostasis. But in the event of a loss of tension trigger, something along the lines of overuse or microdamage, we're going to see a loss of that extracellular matrix tension, which then the cell doesn't really have as many proteins to cling onto, and it sort of devolves into this circular balled up shape. Now, because of that, we know that YAP one of the primary triggers is mechanical stimuli. And since it can't hang on to each of those proteins, we're going to see YAP inactivation and thus MMP production. So the use of siRNA indicates that a knockout of YAP and TAZ increases levels of MMPs. Now, when this group tested YAP in overexpression, they also saw a decrease in MMPs, which is really incredible because now we're seeing this joint response between YAP and TAZ and this onset of tensional homeostasis loss, which is indicated by MMPs. So to zoom out for a bit, we know that a loss of tensional homeostasis is a proponent of tendinopathy because control of that cytoskeletal tension is really crucial to maintaining cell homeostasis and even fate. And we know that a loss of tension activates catabolic gene programming through the axis of YAP and TAZ. So first of all, why is this occurring? What is the exact role that YAP and TAZ play in this early onset of tendinopathy? Now, to answer this question, we first need to illuminate all aspects of the mechanotransductive pathway. And so to answer this, we first need to ask how we can verify this response that we're seeing in terms of the MMPs and YAP and TAZ. So I'll briefly touch on the two technical methods that we used in order to approach this problem. So first, we wanted to verify using RNA interference. So the way this works is we have the RISC complex with an endonuclease that brings siRNA to complementary mRNA to degrade them. So in our case, we have the mRNA of YAP and TAZ degraded, which basically ceases production of YAP and TAZ as a whole. Now, the orthogonal method we're using here is a biochemical inhibition using this drug called CP1. Essentially, what CP1 does is it inhibits that YAP and T binding partnership. So if you remember from a few slides ago, YAP and TAZ need to bind to a binding partner, such as T, in order to translocate into the nucleus and affect genetic outcomes. So if we're disrupting that partnership, you can tell that YAP isn't going to be able to translocate into the nucleus, and it's not going to elicit its effects. What's also really important to understand is, what is this emphasis on this two-pronged approach? The reason we're doing this is because each of these methods alone has off-target effects on the cells that we're studying. But if we're able to get the same MMP response using both of the methods, we'll be able to be that much more confident in our results. So now a little bit about the systematic methods we use. We started off with primary patient-derived tenocytes, which are tendon fibroblasts. We subjected them to either the biochemical or genetic knockdown. We then lysed the cells, isolated them for RNA, made complementary DNA, and ran PCR on them to examine the fold change in different genes that we're interested in. First, we validated the siRNA knockout of YAP and TAZ. So here we see downregulation in both levels of YAP and TAZ in terms of their fold change relative to a scrambled control. We're also seeing a reduction in targets downstream of YAP and TAZ, SIR61 and CTGF. These are basically indicators of cell adhesion, proliferation, growth, and more. Now, in the same samples in which YAP and TAZ were knocked down, we're seeing increases in those MMP levels, which is consistent with those previous investigations on tendinopathy. As for the biochemical knockdown, we're still investigating and troubleshooting this. A key step in this process includes serum starvation. So I'll go into this a bit. Essentially, after we treat the cells, we are putting them in serum-free media. So the cells aren't too happy in this media. They're kind of in this balled up phenotype, which is reminiscent of that one of those first diagrams I showed, in which the cell was existing in the extracellular matrix that had lost its tension. So essentially, what the serum starvation does is it's an in vitro way to model that onset of detensioning. What it also does is it drives down all of the initial YAP and TAZ activity to a baseline low. And so after a while, we replace the media with regular media, and we see this dramatic increase in YAP and TAZ activity. And so since we see this exponential increase, we don't see that, or we expect not to see that in the knockout because of the CP1 treatment. So essentially, we expect to highlight the role of MMP production with YAP and TAZ by seeing a clear difference in between our two groups. And here's just a picture of the tendon fibroblast that I was working in this summer. So throughout this talk, we've touched on catabolic gene programming that occurs through the axis of YAP and TAZ. We attempted to approach this problem using genetic and biochemical methods. And while the biochemical method is still in the process of troubleshooting, it's also expected to contribute to that loss of tension phenotype and an elevation in matrix degradation. And the reason this model is so interesting is because it allows us to hone in on those finer mechanotransductive details of this pathway and helps us understand catabolic gene initiation through YAP deletion. As a bigger picture, understanding the onset of tendinopathy from this molecular approach can hopefully help us devise strategies, preferably non-invasive ones, to better maintain tensional homeostasis. As the program comes to an end, I wish I could have looked at different serum starving concentrations to understand its effects on modulating YAP and TAZ, just to sort of troubleshoot it. And lastly, I'd like to thank each and every member of the Burkle Lab. You guys are so tight-knit and inspiring, and I've learned so much that I know I'll carry on to all of my future endeavors. And I'd also like to thank the CEMB, especially Doctors Amanda, Ariel, and Annie. You guys, thank you so much for hosting such a transformative and life-changing program. With that, I'll open for questions.
Quick question, how long does it take the siRNA to have an effect on these cells?
Yeah, so that's actually something we extensively tested this summer. Initially, we were leaving the siRNA on for 24 hours and then waiting 48 hours to lyse the cells, in which we weren't really seeing the knockdown that we expected. Our most recent experiment, the results that you saw, was we left the siRNA on for 24 hours and then lysed after 24 hours. And so in that experiment, we saw that knockdown.
Our next speaker is going to be Javier Santiago from University of Puerto Rico, Mayagüez. This summer, he was in the Janvey lab. He's going to tell us about biopolymers from hydrogels to tissue engineering. So just give us 1 second.
I vote now. Awesome. Can you hear me now? Awesome. During the course of this presentation, I hope to answer some of the questions that I'm not sharing right now.
This is worse than a Three Stooges comedy. I'm so sorry, everyone. Mhm.
There you go. Awesome. The questions listed here. So most of the work that has been going on during the summer was inspired by previous work in the Legami lab, where they incorporated microparticles into different fiber networks. And what they observed was that by incorporating these particles, they could mimic more closely the mechanical properties of different tissues. And the significance of this is that previously, when you made this fiber networks, the physical elasticity of the properties of the hydrogel would not match with the tissue properties. And so normally what you expect to see from tissue is that as you compress it, it stiffens. But the problem was that when you use hydrogels, it normally compression soften. And so what we're trying to do is incorporate different microbeads in the mammalian cell range of sizes and also in their elastic properties. And so we're making different sizes of polyacrylamide beads and also different stiffnesses. So I encourage you to stick around because Caroline's going to talk more about the stiffness variations. To make these particles, what we do, we have different setups. So we're trying to make different sizes, right? In the top side, we're using a homogenizing setup. In the bottom side, we're using a steering. What we're trying to make is different inverse microemulsions for inside the flask. We have an organic phase with cyclohexane and span85 as a surfactant. And then from the top, we're adding our polar phase. So we have an acrylamide monomer, bisacrylamide cross linkers, TMED catalyzer, and APS for the initiator. And this reaction is a radical reaction, so we need a nitrogen atmosphere to remove all the oxygen that could stop our reaction. So after that, we move it into a centrifuge tube and pellet it, remove the original phase that it was in because it was an organic phase, and we switch it to a more suitable tris. So we do 3 steps, and as the end result, we end with these particles. So you get an idea of how the setup actually looks. In the beginning, we only have the organic phase, and once we start adding dropwise the polar phase, the reaction carries on, and so you can see the actual change and sort of visually track how your reaction is proceeding. But how do you know the sizes of the particles, right? We did microscopy on them for the homogenizing setup and for the steering setup, and we found that when you use the homogenizer, you get smaller particles. So these particles are in the red blood cell range, basically 5 microns. Whereas when you use the steering bar, you get particles in the cell range of adipocytes, so in the 58 micron range. This is important because we want to try to mimic the different cells that might be incorporated into the hydrogels. Then once we have the particles, we introduce them into the fiber network. What we have is on the top, we mix together our buffer, including the particles. Then we add the fibrinogen, which will be transformed into fibrin by adding thrombin in the bottom tube. So we mix everything together, and then we add it to thrombin, and that allows us to create this network with the particles already embedded in them. And what you can see here is different particles just incorporated into our fiber network. So we have a lot of them in this picture. And here I'm comparing the different sizes that we can embed into the network. So on the left side, you're seeing the small particles already in the fibrin gel, whereas on the right side, you're seeing the larger particles in this network. So after we do that, we do rheology. So we compress the hydrogels and evaluate their elastic and their viscous behavior. And what we found, we tried this for different volume fractions. So we did 10, 20, 40, and 60% volume fractions. And also small and large particles. So what we're seeing on the left is basically comparing the effect that size, small or large, at the same concentration will have on the elastic properties of this gel. And so when we use larger particles, you see that the stiffening effect occurs to a greater extent when compared with the smaller particles. Similarly, when you use a larger volume fraction, you end up with a greater stiffening result. But I want to bring your attention into this line in the middle. The green line is basically the small particles at a 60% volume fraction, whereas the other line that's sort of overlapping it is the larger particles at 20% volume fraction. So you can see that you can gain a similar effect in the stiffening properties by using different concentrations just by varying the size of the particles. And this is pretty significant because then you can fine-tune the properties of the hydrogel that you want to use and use the proportion or volume fractions of the reagents and proportions that are adequate for your stiffening desires. So a similar effect was observed for viscous behavior. But I want to bring your attention into how they look under the microscope, right? Because we're seeing a stiffening, but we can also see it qualitatively. On the left side, you have the uncompressed gel, whereas on the right side, you're going to see the gel at 80% compression. And what you can see here is that it is initially relaxed, but as you start compressing, you can see this effect at as low compressions as 20%, 40%. But here's the most drastic effect. You can see that the fibers tend to align in this direction. And so the way I think about this is when you stretch a rope, you get a greater tension, right? So in the same way that we're seeing the stiffening effect here, we can also visually confirm it using microscopy. And we were interested in how particles also move within this network, right? Because you compress it and you can see that the mesh is sort of aligning, but how the particles move within it. And so what I'm showing here is the fibrin gel with a microparticle incorporated in the middle. That's why you can't see anything in the middle. And the smaller particles are actually 200 nanometers fluorescent beads. And what you're seeing there is fluorescent Brownian motion. And we did some image analysis on them to show, to track the paths of these particles, but you don't get a lot with these tools, only using imaging. So I went on a little side project and developed a Python program that could yield more information on how the particles move by giving me the velocity between each point and their position. So you can see like the particle starts here and then it moves to the left with a certain velocity. And that gives you like more qualitative, more data that you can actually quantify to see how the beads move within the network. And so what we saw is that as you increase the compression, the velocity and displacement of these particles tends to reduce lower. So when you start with no compression, you see there's a lot of movement, but as you go down, as you compress more and more, there's less movement. And so we're able to quantify that data using this software. And here, I'm just trying to show you all the capabilities of the program. So we're able to track the average velocity, the displacement, and the total distance traveled by each of these particles, both individually or in the bulk. And so some conclusions from these projects is where that depending on the method that you use to mix the particles, you can get different sizes. So if you want to generate smaller particles, you might want to use homogenizing setup. Whereas if you want larger particles, you want to use the steering setup. We also saw that volume fraction and polyacrylamide size increase the elastic and viscous moduli, and that particle freedom decreases with the compression of the network. So as for future steps, we would like to incorporate living cells into the biopolymeric networks to evaluate their effect, and also develop materials with tunable viscoelastic properties, because this can be used for stem cell differentiation, also for tissue engineering applications. And we would also like to evaluate the stiffnesses and the effect of stiffness and shapes within the viscoelastic behavior of this network. So I invite you to stick around because Caroline's going to talk more about this stiffness effect. I would like to acknowledge everyone in the lab. They were awesome, and this work would have not been possible without their help. Also the people from the CMB, Amanda, Ariel, Annie, you're awesome. And also my mates from the LRSM, we all worked together this summer. And thank you. Questions? Yeah.
So great job, that was wonderful. I was wondering if you could comment on the relative contributions, you talked about Brownian motion. And how does the compression affect that versus convective transport versus sort of confinement within the matrix?
Okay, so if I understand your question, you want to see first the effect on Brownian motion. What we saw was that as you compress the network, so if you compress it and immediately you can see bulk movement. So some particles are sticking to the network, but other are not. So as you compress it, there is less and less Brownian motion. And for convective behaviors like the movement across the network, what we saw is that depending on where you compress it in the gel, so if you're compressing lower, you can actually see the direction changing. And then the other part of the question was
Matrix confinement, so as sort of the matrix compresses.
So the
small.
yeah. So the particles moved like to a lesser extent under compression. So you can, I believe there's a mesh reduction size.
Which of these do you think is the most important?
Right now, I wouldn't be able to like specifically determine one parameter. I would say that definitely the mesh size would be an important factor because we're using a specific size of beads. I'm not sure if that's one part that we're trying to determine the effect of, if the particles are sticky, would they move differently? We also pagulated the surface of these nanometric fluorescent beads to avoid non-selective binding to the fiber network. So what you're seeing is mainly due to the effects of the space in which they're able to move in, and not if they're actually sticking to the network.
That's a good question, but do you think charged particles would act differently?
Yeah, that was one of the questions that I wanted to investigate during the summer, but there's not enough time. But I would expect that if the electrostatic interaction between the beads and the fiber network were favorable, they would move more, or if they're attractive, then they would stick more to the network. So that I expect that to have a change, but that hasn't been explored yet.
So our next speaker is going to be Alex Goodwin from New Jersey Institute of Technology. This summer, Alex was in the Morchiotti lab studying muscle regeneration defects and heterotopic bone formation in fibrodysplasia ossificans progressiva.
You said my name is Alex Goodwin. I was in the Morchiotti lab and my mentor was Lorenz, and we were studying muscle regeneration defects and heterotopic formation in FOP.
Again, to share the screen.
Okay, so our lab studies FOP, and it is a rare genetic disorder that occurs in 1 in every 1 million, and it is characterized by this extraskeletal bone formation, which you see on the skeleton here. Now, this disease is caused by a mutation in the ACVR1, with the most common being the R206 mutation. Now, this is a bone morphogenic protein, which plays a large role in the BMP downstream signaling, which you can see in the center of that far right picture. Now, as a result of the mutation, it is causing hyperactivity within the BMP signaling, which is further driving this HO formation. And it has been found that this is driven by FAPs. So FAPs are cells that reside within the skeletal muscle, and upon injury, they become activated and start fibroproliferating. During this time, there's a crosstalk between the FAPs and the muscle stem cells, which allow them to further proliferate, differentiate, and finally fuse the muscle fibers. Additionally, M1 macrophages come in and they release this TNF alpha, which is causing apoptosis among the FAPs so that they don't overproduce and can further differentiate into fibroblasts as well as adipocytes, finally giving us healthy regenerated muscle. Now, these FAPs, which I've been talking about, are fibroadipogenic progenitor cells that reside within the skeletal muscle, and they have this ability to differentiate into different cell types. However, our lab is most focused on the osteocytes and chondrocytes. FAPs have these different markers, but we are mainly focused on the PDGFR alpha marker, and we found that when it is bound with the Cre system, you see a lot of that HO formation as indicated by these white arrows here. The Cre is a mouse model that occurs from development, and it is slightly different from the one that we use, but I'll be discussing that later. So while there's a lot of previous study on this heterotopic bone tissue, the R206 mutation, which our lab studies, has not had a lot of research done on it. So as we know, there's impairment in the muscle regeneration within FOP, which you can't really see between the knockout and the uninjured in the from day 0. You start to see a little bit more of the divide within the blue and red bars at 5 DPI, and then you see more of the split at 10. And this trend continues into 21 DPI, where you also have this cartilage and bone formation as pictured here. Now, these mutant FAPs, you have the same process where they start fibroproliferating upon muscle injury. However, the difference is that there's an impairment between that crosstalk of FAPs and muscle stem cells. And additionally, when there's the apoptosis, the FAPs are persisting through and are not dying in the way that they should, which causes them to go through chondrogenesis, forming cartilage, rather than fibroblasts and adipocytes. And the difference here that we see is that it's forming the HO, and there's that impairment within the muscle, which is different from before. So during the summer, we had some research goals. We wanted to study FAPs and the role that they play in the microenvironment as it leads to HO. Therefore, my mentor created this FOP mouse model that's aimed at targeting the FAPs within the skeletal muscle. And therefore, I wanted to confirm that this mouse model does work and that it is forming the HO in a similar manner to the way that it would in patients. Additionally, we want to confirm that the tamoxifen is not affecting the skeletal muscle. Finally, we wanted to study this at different time points. So here we have our mouse model where we're using the CRE2 system, which is binding to tamoxifen, and it goes in and it clips the gene, allowing the mouse to express the mutation. And again, just those, we're targeting those FAPs. So for our design, we've injected the mice with tamoxifen every 2 to 3 days, and then we induce the muscle injury in the gastroc and the tibialis anterior. Then we sacrifice some of these different time points. Once they've been sacrificed, we put them through micro CT analysis, which gives us a 3D visualization of the mineralized bone. However, since this is something the cells go through chondrogenesis, we need to study them further using histology. So we section them at these different lines and we stain them, which allows us to further quantify them. Here, just showing that our PDGFR alpha model is working and we are getting that recombination in the DNA. Here are some of the micro CT images that we've gathered over the summer. So as you can see, there's no HO formation in any of the uninjured 3, 5, 7 DPI. You only really start to get it here at 10 and then more at 14. So we've confirmed both of those things, but then we want to obviously investigate the tamoxifen. And we see that when comparing the 3 of these images, the muscle fiber size is all relatively the same. There is a slight decrease with the control plus TAM. However, it's not enough to be statistically significant. Therefore, it does not alter the muscle tissue in any way. Finally, we want to study this at different time points. So as you can see in the uninjured control in our FOP model, there is not much of a change, but you start to see a little bit of it here at 5 DPI with some centrally nucleated muscle fibers. At 10 DPI is when you really start to see more of that split because the control is obviously starting to repair and those fibers are growing back to their normal shape. Whereas the PDGFR alpha FOP model, the size is much smaller. This trend continues with 14 DPI with there being the split in the graph and in the size. We are also actively studying the 21 DPI, but hypothesized to get very similar results as to what we've been seeing already. In conclusion, we've confirmed that there is HO formation in our PDGFR alpha ACVR1 R206H mouse. We've confirmed that the tamoxifen is not affecting the muscle homeostasis in any way, and that there's this FAP-driven impairment of the muscle regeneration, therefore showing that FAPs play this pivotal role in the repair process overall. Looking ahead to the future, we want to continue to measure the muscle stiffness of these fiber proliferative areas within the our mouse. And we want to further understand what is going on early on in the stages with these FAPs and what is leading them to misinterpret their environment, further causing HO. Finally, I'd like to give my thanks to my mentor, Lorelis, and Dr. Morchioti for supporting me during these 10 weeks. It has been truly a pleasure. I also want to thank the CEMB for hosting this lovely program, truly transformative. And finally, I'd like to give my thanks to my PI back home, Dr. Alex Buffon, for supporting me during my application process and all of my previous research experience. Thank you. Any questions? Yeah.
So, in patients with FOP, the mutation shows up at birth, right? So, can you talk about why you've chosen this inducible approach and what might happen if you were to do, rather than the tamoxifen inducible, if you were to have a constitutive activation of that mutation?
We've kind of done that with the Cree. The Cree, we use like younger mice, but we see that there's like much higher death rates. And it's very, very, like the HO formation is like more persistent and there's a lot more growth more quickly. So our model allows us to study this like for longer time periods.
Wonderful. So our next speaker is going to be Caroline O'Rourke from the College of New Jersey. In the summer, Caroline was in the JMA lab. And today, Caroline's going to tell us about the effect of polyacrylamide microparticle stiffness on fibrin hydrogel rheological behavior.
Hello everyone, my name is Caroline. I'm a rising senior at the College of New Jersey, and this summer I worked with Dr. Janney's lab to determine the viscoelastic behavior of fibrin hydrogels containing specifically stiff microparticles. So here's an outline of what I will go through today with an introduction talking about the significance of hydrogels, why we want to improve them, our methods for doing so, and the results and conclusions that we obtained. So ultimately, we are developing biomaterials, which are any kind of material that supports biological function. And these may be types of implants or prosthetics, but ultimately, in order for these to actually be effective, they need to be compatible with our bodies. So for example, although this Band-Aid over here might work very well at wound healing, it may work better if it more closely resembled the properties of living tissue. And so my project focused on hydrogels, since our bodies produce these naturally, such as collagen for support and fibrin in blood clotting. They are excellent candidates for making a biocompatible hydrogel, which can be used for all sorts of medicinal purposes. And we are going to be using fibrin to make the hydrogels in our lab. Again, hydrogels are a type of polymeric biomaterial, meaning that they're made up of repeating units that cross-link to produce a matrix. So if we just isolate the important proteins involved in that, we can make a gel. And so the way that this works with fibrin is that fibrinogen exists inactive in the blood until an injury to the blood vessel signals thrombin to leave these fibrinopeptides and allows a chain reaction to take place where all of these monomers coagulate into a matrix to clot the injured vessel. And here is the macro gel-like structure of what eventually formed, which is great since now we have an amazing hydrogel that can be used in bioengineering. But we are forgetting something, that hydrogel doesn't really mimic the properties of living tissue, which is so important for being considered a useful biomaterial. So why does the gel not equal real tissue? Because in real tissue, there are many different types of cells, such as red blood cells and platelets, that are situated in between all of those fibrin polymers, which make up something called the extracellular matrix in the human body. And they are volume conserving, meaning that when you squish them, they generally retain the same amount of fluid. So this means that when the overall tissue is compressed, it actually stiffens. And this compression stiffening is denoted here by the red line. This graph is just a summary of previous findings by the Genulab that when tissue is compressed, its elastic modulus, which we denote as G prime, increases, meaning that it does become stiffer. However, plain hydrogels in the blue line are mechanically the opposite of living cells. When a hydrogel itself is compressed, its stiffness stays the same. But previous experiments shown by the purple line did find that adding particles helps the gel to follow the same type of mechanical trend that we see in living tissue. However, the reason for why this occurs is still undiscovered. And so our hypothesis is that specifically stiffer particles in the fibrin network is the reason for this trend. So first, we made our particles out of polyacrylamide using inverse emulsification, and we achieved a group of particles with higher stiffness by using a higher concentration of the crosslinker bisacrylamide. And we can visualize them under the microscope to confirm uniform size and shape. So here they are in just PBS solution at 10x and 40x magnification. And then we ultimately put the particles in the gel, again, which was made using all of these ingredients involved in fibrin coagulation, and our particles, which were stored in tris buffer. And these pictures are not to scale, but here is theoretically what it looks like in the gel now, with the particles now situated in the spaces between the network. And here are the particles in the actual gel visualized under the microscope. Again, going back to this picture of a blood clot, the function of the particles is to act in place of all these cells. And then we characterize the mechanical properties using stepwise compression testing on a rheometer. So we put the gel on this plate in the center, and this flat piece is completely parallel with the plate and can compress the gel up to 60% and measure its elastic modulus at each compression point. So as compression increases with each step, the gel deforms slightly and causes the fibrin network to respond in some way, which we can determine through how the elastic modulus changes. And so here are the results of the compression testing. Each group was measured at 10, 40, and 60% volume fractions. So for example, 60% of the gel that we made was particles, and a control which had no particles at all. Again, G prime is a measure of the elastic modulus or the response of the elastic part of the network, not the viscous part, to deformation. So an increase in G prime would indicate that the network itself is becoming more rigid. So when the control in both of these is compressed, the G prime initially decreases and then stabilizes as compression percentage increases, which is expected since polymer networks by themselves are known to soften in compression due to when the individual polymers buckle on each other, the network collapses. But for soft particles specifically, as the volume fraction increases, the G prime also increases with compression, which is attributed to the fact that more particles in the hydrogel actually does the opposite. It prevents the fibers from buckling and stops the network from collapsing. Interestingly, we do not see this dramatic compression stiffening from the stiff particles until about 60% volume fraction, which is at the point where there are just so many particles in the gel that it's really the amount and not the particle stiffness that is causing this change. But as far as 10 and 40% not increasing their elastic modulus too much, some proposed explanations is that the particles may not really be attached to the network. So when it's compressed, the particles move and it appears as though the gel actually has no particles in it. So overall, softer particles cause the hydrogel to become more rigid under compression, which is more consistent with the behavior of living tissue that we're looking for. And so overall, we concluded that particle stiffness specifically may not be the reason for compression stiffening of hydrogels with particles. And a characteristic that we are looking forward to testing next is how differently shaped particles change the mechanical properties. So we know that real cells are absolutely not perfectly spherical like our particles were. So an elongated particle may better attach to the network and therefore strengthen the stiffening on compression and prevent the fibers from collapsing. And next, we concluded that since stiff particles do not cause dramatic compression stiffening and soft particles do, that softer particles are more consistent with the mechanical properties of living tissue and therefore might be better to use if we want to make biocompatible hydrogels. But ideally, real cells would also be added to obtain a more biologically accurate measurement, since real tissues have a higher volume fraction of cells than what we were able to add with particles. So thank you to everyone in the JAMI lab for your mentorship this summer and everyone from the CEMB for coordinating this program, and also to Michaela and Torin, who were our lab mates from the LRSM program. And that's it.
Okay. Hey, everybody. So our next speaker is going to be Winnie Maru from Bryn Mawr College. And this summer, Winnie was in the Disher lab. And today, Winnie's going to tell us about exploring genomic instability in cancer, how mitotic perturbations lead to chromosome missegregation and loss.
Hello, everyone. So like Dr. Parker said, this is the talk I'll be giving, exploring genomic instability in cancer. How mitotic perturbations lead to chromosome segregation and loss. So there are a number of factors that contribute to cancer's tumorigenesis. Sustained proliferative signaling, as you can see up here, leads to uncontrollable cell division, and genome instability and mutation may contribute to cancer's initiation and progression. So chromosome errors usually occur, chromosome segregation errors usually occur in mitosis, and these are the different ways that this can occur. So incorrect attachment of spindles to chromosomes, which can lead to chromosome fragmentation in the form of a micronucleus, and also other large-scale chromosome events such as deletions, duplications, inversions, substitutions, and translocations. So these chromosome losses and gains and rearrangements can confer cancer cells with a growth and migration advantage and allow them to proliferate despite various immune cells and also immune activity, and also spread to secondary sites. So chromosome losses and gains are frequent in solid tumors. As you can see here on the left, acute myeloid leukemia is a liquid cancer of the blood. So you don't see any chromosome losses or gains. However, in solid tumor types, you do see these chromosome gains and losses. And in my lab, we worked with cell lines derived from breast invasive carcinoma, lung adenocarcinoma, and melanoma. And you can see that they do have these 3 different tumor types do show increased chromosome loss. And over here, you can see a proportion. So this is a proportion of tumors that display chromosome losses or gains in within a given chromosome. So mitosis is a very regulated process controlled largely by the activity of cyclins and cyclin-dependent kinases. And these 3 genes are important throughout the mitotic process, and their dysregulation has been shown to contribute to cancer's phenotype. So MPS1 kinase is part of the spindle assembly checkpoint that ensures that chromosomes, that ensures bipolar attachment of chromosomes along the mitotic spindle. So you can see it here localized within the nucleus, and here it's shown in yellow fluorescent protein, and it's localized with the with the centromeres in red. And PTEN is a phosphatase that is known for its regulation of the PI3K-AKT pathway, and it negatively regulates this pathway, which is involved in cell proliferation. So it works as it is a putative tumor suppressor. And recent studies have also found that lack of PTEN, that actually PTEN localizes with a key centromere protein CNP5, and loss of PTEN leads to chromosome fragmentation and also some Robertsonian translocations. And myosin 2A is a cytoskeletal motor protein, and it is involved in formation of the contractile ring in cytokinesis. So up here you can see this is the nucleus. This is a DNA stained in blue with DAPI, and then the green is aniline, which is a microtubule protein, and within you can kind of see small red specks, which is the myosin 2A. So it helps to form this contractile ring, and this is in late stage cytokinesis. So we aim to pharmacologically inhibit these 3 drugs to observe any changes in nuclear morphology in the MCF7 cell line. So this is the MCF7 cell line in low density and high density. It usually forms these clumps, and it is a estrogen receptor positive breast cancer, which is what most of the breast cancers are. Estrogen ER positive. So this is our experimental setup. We had VO-OPIC, which is a potent PTEN inhibitor, and blagstatin, which inhibits myosin, the gene for myosin 2A, which is MYH9, and reversing, which is a reversible MPS1 inhibitor. And this treatment was performed for 24 hours without washing the drug out, and we fixed the cells and permeolized them and immunostained and performed fluorescence microscopy. So here we observed mitotic cells in each of the conditions, and also micronuclei. As you can see here, it's contained within the F-actin phalloidin. So as you can see, and what we found was that there was, with reversing, There was increased micronuclei formation with decreased mitosis. And with blebbistatin and P10 inhibition, there was increased mitosis with increased micronuclei, though blebbistatin's result was not significant in this case. So this makes sense for blebbistatin and P10 because P10 is a tumor suppressor. So as we inhibit this tumor suppressor, we get increased proliferation. And the literature has some support for MYH9 being a tumor suppressor. And in this case, our data may suggest that that is the case. The evidence is kind of, MYH9 is, some literature says that it is a tumor suppressor, some says it's not. But here we can see that when we inhibit it, we get increased proliferation. And like I said here, it seems like reversing is reducing, is causing increased micronuclei with less division. And we would expect that as more divisions happen, we would get increased mitosis, increased micronuclei. Though since reversing is affecting the spindle assembly checkpoint, it may be stressing the cells so that they're not able to divide, but they have other stressors that are causing them to have this nuclear morphology changes in the form of a micronucleus. So we also chose to look at a breast cancer treatment drug called palbociclib, which is a CDK4/6 inhibitor. So it targets molecules, the CDK4/6 enzymes within mitosis. And it specifically prevents cells from going through the G1 to S checkpoint. And we have, so there's some evidence that this drug, although it's very potent against breast cancer, it causes some genotoxic stress, which is unexpected and needs to be further studied. So we see that when cells are, this paper talked about how when cells endure prolonged G1 arrest, they may continue, most of them exit the cell cycle in a p53 dependent manner. However, those cells that are p53 null may continue through G2 and into mitosis and experience a catastrophic mitosis event leading to chromosomal missegregation. And as we know, p53 is usually mutated in cancer. So this is an area that needs to be explored, whether this drug is causing genotoxic stress. So we did this experiment, similar setup to the previous one, where we just used 10 micromolar of palbociclib on the MCF7s for 48 hours. And this is our control treatment, DMSO. So we did observe some micronuclei, and also in the palbociclib, we observed some micronuclei formation. So our result was not significant. However, this was only one replicate, and more replicates may be able to show that increased micronuclei with palbociclib is significant. And we also saw increased nuclear blebbing, and nuclear blebbing can be a sign of nuclear fragility, because the nuclear envelope is kind of blebbing and is showing to be unstable. So we then chose to look at, we then continued this project in the lab, which is aiming to create chromosome reporters, live cell chromosome reporters to track chromosome loss. So we decided to look at adding this reporter to 3 constitutive genes. So these are genes that are always expressed in the cell, and we chose these because these genes localize to a particular point. So when we do the microscopy, we are kind of confident that it's not autofluorescence, like the nuclear lamin localizes to the nuclear envelope, and we can use DAPI and also see so it's not autofluorescence. So how we do this process is we have our gene of interest that we want to add the fluorescent tag to, and we have our plasmid with the green fluorescent protein, and we add that to the beginning of the gene, so it's an endogenous gene tagging, and we use a monoallelic approach, so tagging only one allele of the chromosome we're interested in. So this is what we did. So we obtained the plasmid from E. coli and we inoculated into some media and we let that grow for about 24 hours. We then spin it down, aliquot it and spin it down to obtain a pellet, and then resuspend the pellet in some fresh media, and then do a spin column to isolate the DNA plasmid. And we use the nanodrop to measure the DNA concentration and the purity. And this is the CRISPR lipofectomy method of how we're going to introduce this gene into the cells. So we use the CRISPR RNA and the tracer RNA, we combine those plus the Cas9 to create the Cas9 complex here. And we add the donor plasmid. So this is going to tell the cell where to cut, tell the enzyme where to cut, and then gonna add the plasmid in. We did it in murine B16 cells, which is a melanoma, derived from melanoma, and also MCF7, which is human breast cancer. And then we wait a few days, few weeks, depending on like the growth rate of the particular cell line. And we isolate green colonies, which are going to be rare events, and then do some sorting to isolate them and continue to grow them. So here you can see we attached the chromosome reporter to lamin B1. So here we can see GFP lamin B1 in murine melanoma signals increased micronuclei formation despite decreased cell division. And here we observed most of the population of this cell, these cells were GFP positive. However, we did note some GFP negatives. This is how a mitotic cell looks like, and micronuclei, and we also observed nuclear blebbing. So conclusions. So when we inhibit key mitotic proteins, it leads to key mitotic genes, it leads to abnormal nuclear morphology. And we can see that we have evidence that mitosis is a key step that leads to chromosomal instability. And we can use a live cell chromosome reporter method to measure chromosome loss under a variety of stressors. And future directions is we'd like to recapitulate the in vivo tumor microenvironment. And one way we can do this is doing confined mitosis to observe changes in nuclear morphology and also spheroid formation. Since we've seen that MCF7s, it's been noted that MCF7s display drug resistance when they're in a tumoroid form. So yeah, thank you. The video was not playing, but this was going to show the squashing experiment with the confined mitosis. And those are my references. And thank you to the Disher Lab and the CEMB program.
So our next speaker is going to be Ikra Sajid from Alabama State University. And this summer, Ikra was in the binding lab. And today she'll tell us about manipulation of immune response through IL-2 and B-cell acute lymphoblastic leukemia.
Good morning. As Ariel said, I had the opportunity to work in the binding lab this summer, and so I researched the manipulation of immune response through IL-2. I'm going to start off with a table of contents, kind of just outlining everything that I'm going to be talking about today. My motivation, a background on T cells and B all cells, IL-2, the results, conclusion, and some future directions. So how exactly does what I was doing over the summer connect with mechanobiology? One of the biggest things is bone marrow fibrosis. So B-all has a low response to CAR T therapy when the patient also suffers from bone marrow fibrosis. Bone marrow fibrosis is a lesion that occurs when reticulin, a protein that represents collagen, and the collagen fibers increase or proliferate. In the bone marrow, it changes the mechanical properties of the environment. It also is associated with changes in the permeability of cytokines, and it also potentially creates a local immunosuppressive environment. Some background on T cells. So T cells are a type of white blood cell that develops from stem cells in the bone marrow. It also identifies invaders like viruses and bacteria to help control the overall immune response. It also helps activate cancer and B cells to produce antibodies and send signals to coordinate the immune response. What are B-all cells? B-all is the cancer of the blood in the bone marrow that originates from immature B lymphocytes, which are supposed to develop into cells that produce antibodies. In B-all, these immature B cells, also called lymphoblasts, proliferate uncontrollably and do not mature properly, leading to an accumulation of dysfunctional cells in the bone marrow and blood. What is IL-2 and its effect on T cells and B cells? So when a T cell receptor recognizes an antigen, IL-2 is produced as a part of the activation process. It mainly affects T cells and making them activate, helping them grow, survive, and develop. Because of its strong ability to boost the immune system, IL-2 is used in treatments for cancer and may also help manage autoimmune diseases. IL-2 is a cytokine that helps the immune system communicate and work properly. It enhances T cell cytotoxicity, aiding cancer treatments like high-dose IL-2 therapy, CAR T cells, and engineered IL-2 variants for better anti-tumor responses. IL-2 can stimulate the proliferation of B-all cells, and it also acts as a growth factor, promoting the expansion of these cancerous cells. It can also enhance the survival of B-all cells by activating signaling pathways that prevent programmed cell death. Now on to some of the results. So the first experiment we did was the effect of seeding density on B-all cells and their expansion. So we kind of assumed that the higher the seeding density was, the higher the expansion rate it was going to be, and that's exactly what happened. These are some images I took from the microscope. So as you can see, the lower seeding density has more sparse areas, while the higher is a lot more crowded. Then we explored the effect of IL-2 on B-all cell proliferation in the presence of 9 non-cytotoxic T cells. So this is kind of an experimental workflow. We had an effector target ratio 10:1. So the effector cells were the T cells and the target cells were the B cells. We spun down the cells and collected the media, and then we added a lysis buffer to lysis cells, which is basically a process where we break open the cells to basically kill them and release everything within the cell into the solution. Then we added luciferin, and when luciferin reacts with luciferase, it produces the luminescence, which makes the B cells glow. And based on how much luminescence we get out of the reaction, we can determine how many B-all cells are there. So here on the left, this is just a product of just pure B cells with IL-2 without any T cells. So it kind of works as a control. And then on the right, we have a co-culture system with T cells, B cells, and IL-2. The red at the end is just pure B cells. So you can kind of compare that to the dark blue on this side. Then we quantified the amount of interferon gamma that's being released. So interferon gamma is a cytokine associated with T cell activation and cytotoxicity. Originally, we were expecting a reverse trend from the luminescence assay, but however, due to an extremely low cell seeding number, the interferon gamma release is pretty low. So there isn't very like a common trend, and we think that's because of the low seeding number. For my conclusion, the higher IL-2 the concentration means both of the higher T cell proliferation and B cell proliferation. The endogenous T cells, so just pure T cells, don't really have that much of a targeting effect on B all. However, they do produce more IL-2 with the induction of the IL-2 feedback loop. So kind of tying back into the beginning, more IL-2 means more B all cells. For some future directions, we can quantify the amount of IL-2 release with ELISA, and we can also use the hydrogel system to encapsulate these cells in 3D, because basically everything we've done is in 2D. And my references. And I just want to say thank you to my mentor, John Chen and Nini Tran, and Dr. Vining, as well as everybody in the Vining lab that helped me throughout the summer. And of course, obviously the CEMB program. I wouldn't be here this summer without everybody. Thank you. Questions?
Can you explain the IL-2 feedback loop briefly?
Okay, so that feedback loop is basically a cycle where immune cells can stimulate their own like proliferation and activation in the production of IL-2.
So our next speaker is going to be Maya Taggart-Geis from Cornell University. And this summer, Maya was in the mock lab. And so Maya's going to tell us about the effects of pathogen-associated molecular patterns on synovial fibroblast mechanosensitivity. So just give me a second. Oh.
All right, hi everyone. My name is Maya, as Ariel said, and this summer I had the pleasure of working in the mock lab. And today I'm going to talk about my project, the effect of pathogen-associated molecular patterns on synovial fibroblast mechanosensitivity. So osteoarthritis of the knee joint is a very common disease which impacts about half of people throughout their lifetime, and it can be characterized by cartilage degradation, joint gap narrowing, and synovitis, which is inflammation of the synovial membrane. And this membrane lines the joint cavity and secretes essential joint lubricant. And chronic synovitis is often associated with synovial fibrosis, which is what this research will address, and is also associated with major symptoms of OA progression like joint pain and stiffness. So one of the cell types within the synovium are fibroblast-like synoviocytes, and these are responsible for secreting joint lubricant and collagen for the ECM. And when they become activated to a myofibroblast phenotype, they become increasingly contractile, decrease lubrication production, and increase collagen production. So here, excess ECM deposition creates a stiff environment that triggers their mechanoactivation. And the myofibroblast phenotype is therefore associated with fibrosis and the progression of osteoarthritis. So it has already been shown that biophysical cues like substrate stiffness can in fact alter fibroblast phenotype. So here, as substrate stiffness increased, so do characteristics of mechanoactivation. On the top, you can see that cell area increased as cells became more contractile and spread out, and that's also depicted in the graph right there. Next, we see alpha smooth methyl actin, which is a type of actin that supports F-actin when under stress, also becomes increased with stiffness. And lastly, as cells become more contractile, they form more focal adhesions, which activates transcription factors associated with YAP and TAS. But the question still remains, why fibroblasts act poorly in osteoarthritis before any physical changes occurred in the synovium? We hypothesized biochemical cues as well as biophysical cues synergize to trigger the inflammatory response that activates these synovial fibroblasts. So the innate immune response is activated by two types of signals. Damage and pathogen-associated molecular pattern, or DAMPs and PAMPs as I will now refer to them. So these interact with toll-like receptors, or TLRs, on the cell's surface, and here we aim to explore the impact of PAMP-mediated inflammatory response on fibroblast mechanoactivation. So to recap, the link between cell mechanoactivation and physical environment is widely studied. However, the introduction of lipopolysaccharide, a TLR4-inducing inflammatory signal, to fibroblasts on tissue-like substrates can in fact explore the impact of PAMP-mediated inflammatory response on cell mechanoactivation. So our research investigates this relationship between inflammation and mechanoactivation, which is a novel approach to understanding the mechanisms of synovial fibrosis. Here we use synovial fibroblasts isolated from bovine knee joints. We did primary culture for 24 hours, and then we passed cells twice before usage. To plate cells, we utilize a culture system mimicking ECM stiffening during fibrosis, and we use polyacrylamide hydrogels with stiffness moduli of healthy and fibrotic tissue, so 5 and 55 kilopascals. We then introduce LPF at the time of cell seeding. And then after 3 days of incubation, we fixed cells and we did immunofluorescence imaging for ASMA, and we did counterstaining with fluorine and Hoist. So to analyze cell shape, we use the following parameters. First, we did cell area because we know that cells tend to spread when contractile. We looked at cell aspect ratio, which is the cell's major axis length divided by the minor axis length. And we know contractile cells tend to be more striated than round. And lastly, we looked at solidity, which is the cell area divided by the area of its convex hull. And we know a circular cell would have a solidity closer to 1, whereas a contractile cell would have a solidity closer to 0. So after imaging, it seems that cell area did increase and solidity decreased as the cell environment stiffens, which is what we would expect. But in the presence of an inflammatory signal, the trend in area and solidity is a lot unclear. So further analysis verified that synovial fibroblast phenotype can in fact be altered by exposure to physical stimuli, and cell solidity significantly decreased between soft and stiff substrates for cells without LPS. And cell area appeared to increase between soft and stiff substrates for cells without LPS as well. And there's no significant difference in cell aspect ratio. So some of the reasons we think we did not see these trends presented as we would expect could be the fact that we had difficulty of cells adhering to our soft gels, and therefore we had a smaller sample size than intended. And additionally, we only used one incubation time point of 3 days. We suspect that a longer incubation time point would be enough time to see mature changes to cell morphology.
But what we found really interesting was the clear difference in ASMA production for cells grown on soft substrates with exposure to LPS. So our 5 kPa gels are analogous to healthy tissue, and with no inflammatory signal, we would expect very few ASMA stress fibers. And this is depicted in the top 2 panels. So as you can see on the soft gels, when we have no inflammatory signal, there is no ASMA stress fiber as expected, but when we introduce the inflammatory signal, there is. And so the significant increase in ASMA expression when LPS is introduced to the cells in healthy tissue-like environments does imply that mechanoactivation is triggered by mechanical cues. So to conclude, while our observations of no change in cell area and aspect ratio were unexpected, they could be explained by our small sample size or potentially our incubation period was not long enough to fully stimulate the change in morphology. And our observations of cell solidity are consistent with what we would expect, where cells become more contractile in stiffer environments. And finally, we imply biochemical cues are capable of mechanoactivation as ASMA increase with exposure to inflammatory signal. It's also important to note the limitations with our study. So as I said, we had difficulty getting cells to adhere to soft gels, and this resulted in a smaller sample size. And we also looked at specific TLR interaction with LPS at only 1 dosage and for 1 incubation time point. So as with all research, our experiment inspired many more questions beyond the one we investigated. So it's been shown that LPS likely interacts with TLR4, and to verify this, we could repeat the experiment with the TLR4 inhibitor, as well as play around with different dosages of LPS and incubation times. We would then verify the signs of mechanoactivation we saw with our immunofluorescence imaging with gene expression for inflammatory and fibrotic markers. We could also repeat the experiment using a DAMP, such as fibronectin fragments, to explore the impact of endogenous inflammatory signals, which are thought to follow similar pathways as PAMPs. And using a DAMP would be a better model for osteoarthritis, and we would analyze mechanoactivation in the same way. Lastly, this experiment could be expanded to an in vivo mouse model where we inhibit TLR signaling and then induce synovial fibrosis. So we could either genetically delete TLR signaling within the mouse genome, or we could develop an injectable therapy to inhibit TLR signaling. So thank you to the Mock Lab and to Dr. Mock and my mentors, Lance and Ellie, for helping me this summer. And thank you to everyone in the CMB for making this possible for me. And at this time, I can take any questions.
Todd Mock said outstanding presentation. Okay, so our next speaker is going to be Daniel Luco from Carnegie Mellon University. And this summer, Daniel was in the binding lab, and Daniel's going to tell us about investigating collagen fiber self-assembly within tunable hydrogel matrices.
Hello, everyone. So for the past 10 weeks, I've been fortunate enough to work with the binding lab at Penn under Professor Kyle Vining and Asal Tavakoli as part of the CMB program. And this is a presentation of my work for this summer, investigating collagen fiber self-assembly within tunable hydrogel matrices. So why do we care about collagen? Collagen is the main component of the human extracellular matrix, and it plays an important role in regulating tumor progression and proliferation. It has been well established that solid tumor cancers change the viscoelasticity of tissue, rendering them stiffer and more fibrotic than surrounding healthy tissue. This is caused by the overproduction of collagen by solid tumor cells. This fibrotic tissue then upregulates tumor cell growth, spreading, and adhesion, as well as impacting further collagen formation and subsequent self-assembly. So to better understand the viscoelastic conditions in which collagen assembly is regulated, a cell-free biomaterial-based approach can be taken, using collagen embedded within 3D hydrogels as an in vitro model for human tissue. This study specifically uses alginate hydrogel, which is a biocompatible biphasic polymer network. What makes this hydrogel a valuable in vitro model is its mechanically tunable nature. Using both unmodified alginate and alginate modified with click chemistry groups, significant modifications to the hydrogel's final mechanical properties can be achieved. So it's the adjustable nature of this hydrogel that makes it an effective platform to study the formation of collagen fibers within different 3D viscoelastic conditions. So we achieved this by using unmodified alginate and alginate modified with click chemistry groups, norbornene or NB and tetrazine or TZ in varying ratios at a concentration of 1.5 weight percentage, mixed with precipitated calcium carbonate and type 1 collagen derived from rat tail. Click chemistry is a term for certain high yield, selective, and spontaneous reactions. Everything was kept at 4C to prevent collagen assembly prior to the gelation of the hydrogel network. How the gelation works is that the alginate forms ionic bonds through cross-linking with the precipitated calcium carbonate in the hydrogel, while norbornene and tetrazine introduce covalent crosslinks to the matrix. So the two methods that we used to quantify the assembly of collagen were SHG imaging and rheometry. To prepare for SHG imaging, the gel samples were plated and kept at 4 degrees C, which, as mentioned before, allowed for the gelation of the hydrogel without any collagen assembly. It was only after the hydrogel matrix was fully formed that the samples were then incubated at 37 degrees C, which then let the collagen assemble. The samples were then imaged using second harmonic generation imaging, which is a type of imaging that lets us selectively look at the collagen fibers. Once imaged, collagen assembly was quantified by calculating the area of the image covered by the assembled collagen fibers. So for rheometry, the gels were again prepared at 4 degrees C and pipetted onto the measuring stage of the rheometer. The samples were then subjected to shear oscillation, time sweep, and frequency sweep testing at 4 degrees C to determine their mechanical properties, such as their storage modulus and tan delta. So by modifying alginate with NB and TZ, a much wider range of viscoelastic conditions can be achieved than when using unmodified alginate hydrogels alone. Figure 1 shows us that the storage modulus, which essentially measures a material's ability to store energy elastically, increased, and tan delta, which quantifies the way in which a material absorbs and dissipates energy, decreased when utilizing alginate modified with NB and TZ, indicating that the gels became stiffer and more solid-like with the higher cross-linking density caused by the introduction of NB and TZ. Notably, tan delta halved between the unmodified alginate samples and the samples that possessed NB and TZ, implying that the covalent cross-linking caused by the norbornene and tetrazine reaction, along with the ionic cross-linking, greatly affected the gel's final viscoelastic behavior. So figure 2 demonstrates that increased cross-link density, which resulted in stiffer, more solid-like gels, fairly impaired the assembly of collagen fibers within the hydrogel matrices. Incorporating the NB and TZ modified alginate, which produced gels with a lower tan delta, decrease the area fraction of collagen fibers, suggesting a correlation between tan delta and collagen fiber assembly. To conclude, by varying ratios of normanine and tetrazine added to alginate hydrogels, several different gel chemistries with biomechanical similarity to native and diseased ECM can be achieved. Embedding collagen within these gels allowed for collagen assembly to be imaged and quantified, from which an apparent correlation between the tan delta of the hydrogel and the magnitude of collagen within the hydrogel was found. This method of using collagen embedded within different hydrogel environments can provide a more effective platform to study the impact of collagen fibers on the growth of tumor cells in further research. This would better enable the in vitro examination of tumor malignancy and progression under various viscoelastic conditions similar to what would be found in vivo. I would like to thank the entire Vining lab, Asal, Professor Vining, the CMB program, Penn Engineering, and Penn Dental Medicine for supporting my research over this summer, and it's been a true pleasure and an honor. And any questions?
A little bit why you did that 4 degrees Celsius?
So most tests were done at 4 degrees C because that's the temperature that allows for the gelation of the hydrogel. Like the cross-linking can still happen, but collagen doesn't assemble at 4 degrees C. So another aspect of the project that we weren't able to get to as thoroughly was we could also do these tests at 37 degrees C as well, which would also allow for collagen assembly. And then we can see how that would affect the final viscoelastic behavior of the gel. But for this part, it's just 4C.
I'm gonna ask, the tetrazine and the ND,
Mhm.
are you the first to have like explored hydrogel behavior with adding those two into the?
I don't believe so. I believe click chemistry groups have been incorporated into hydrogels in the past, but okay. But I honestly I can get back to you on that. I'll make sure before I say anything. I'll make sure like what exactly the most novel part of that is.
The idea that you tune the
Yeah.
viscoelastic properties.
Yeah.
So I see you can extend it to study in vivo, is that what you're saying?
We can extend it to study in vivo, but most of this is the coolest part about this is it's mainly a platform to also just study everything in vitro, where the next steps could be taking tumor cells, which in this case the lab is working with mouse oral carcinoma cells, and encapsulating it within these hydrogel environments, and then studying malignancy progression from there. So everything, much more can be done in vitro now than before, and hydrogels are pretty nice to work with. They're pretty easy to make, and they don't require toxic, expensive ingredients to use. So it's a very nice model to have.
Our next speaker is going to be Kalia Avery, coming to us from Alabama Agricultural and Mechanical University. And this summer, Kalia was in a PO lab. And so today, Kalia is going to tell us about epigenetic responses to fluid shear stress, mechanistic insights into atherosclerosis.
Okay, good morning, everyone. As Ariel said, I am Kalia Avery. I am a rising senior biology major at Alabama Agricultural and Mechanical University. And for my summer project, I researched the epigenetic responses to fluid shear stress to get a better understanding of the mechanistic insights into atherosclerosis. So So first, atherosclerosis is a cardiovascular disease that is characterized by the accumulation of different fats and lipids that come together to build a plaque that kind of lines the blood vessel walls. So as you can see here in this schematic, where there is no plaque, this would be an area of low stress, and where there's a lot of plaque, that would be an area of high stress. So what is FSS and why is it important? Fluid shear stress is a force that acts parallel to a particular surface. So specifically in atherosclerosis, the force would be the turbulent blood flow, and the surface would be the blood vessel walls. But more insights are needed to explore the effects of FSS stress within the cell nucleus, and that's what my project aimed to do. So to do this, we had to study mechanotransduction, which is the process by which mechanical stimuli are then transformed into chemical stimuli. And this is done using G-coupled protein receptors. So GCPRs are an integral transmembrane protein that transmits signals across the cell membrane. They do this by, again, turning mechanical stimuli.
So do you think I'm intruding on your territory?
Why would they do this by turning mechanical stimuli into a chemical stimuli within the intracellular environment. So as you can see in this schematic, as well as the blood flows through, it activates the GCPRs, and they are able to transmit calcium ions into the intracellular environment from the extracellular environment. So the objective of my project was to explore how FSS affects changes within the cell nucleus. We specifically did this by exploring how it regulated different histone modifications and chromatin organizational structure. We also wanted to use FSS to alter GCPR signaling pathways through mechanical cues. So we did this by utilizing an LPA drug treatment to increase cell contractility and thus activate the GCPRs, since it serves as a mechanical stimuli. And then we observed the downstream effects of this. So here's a quick workflow of my project. So on day 0, we seeded 25,000 HeLa cells into a microfluidic device, and then we allowed them to incubate overnight. And then on day 1, we were able to use a mechanical pump to apply force to the cells for 15 minutes. And then for high force, we use 10 dyne for 15 minutes. And for low force, we use 1 dyne for 15 minutes. And then after applying the force, we were able to fix the cells, antibody stain them, and utilize immunofluorescence imaging and super resolution imaging on the second day to quantify our results. So the first thing that we looked at was yes-associated protein, which is also known as YAP. YAP is induced by FSS and involved in contractility changes. So as you can see in this schematic, as the extracellular matrix stiffens and becomes more contractile, YAP is translocated from the cytoplasm to the cellular nucleus. So we thought this would be a good way to view the activation of GCPRs. So in our results, we found that low FSS decreased YAP nuclear localization at 1 dyne for 15 minutes. And you can also see this in our representative immunofluorescence imaging. And a decrease in YAP nuclear translocation is also associated with cellular actin upregulation, a decrease in cellular actin upregulation. So we also added that. In the area of high FSS, YAP nuclear localization was increased, which means that we also saw an increase in cellular actin upregulation here. And we ultimately found that FSS increases YAP nuclear localization. The next thing we looked at was these 2 histone modifications. So the first histone modification that we looked at was histone H3K27 trimethylation, which is associated with gene repression since it has a heterochromatin tag. And the heterochromatin is more condensed, as you can see in this image here. So it is transcriptionally inactive since it is harder to transcribe. We also utilize histone modification H3K9AC, which is associated with gene activation since it has a U chromatin tag, which you can see, and it is less condensed, so it's easier to transcribe and it is transcriptionally active. So for the trimethylated histone at low FSS, we found no change in the mean fluorescence intensity, which means that we didn't find any change in gene repression. But at high FSS, we saw a decrease in the expression of this histone, which means that we saw a decrease in gene repression, which can also be seen on our graph and in our representative images. For histone H3K9AC at low FSS, we saw a decrease in the expression of this, which means that we saw a decrease in gene activation. At high FSS, we also saw a decrease in this histone, which means that we still saw a decrease in gene activation. But if you could reference the graph here, the decrease was still a little bit higher than it was for the low flow group. So at high flow for histone H3K9AC, gene activation wasn't decreased as much at high FSS as it was at low FSS, which basically just told us that we know for a fact that FSS influences histone regulation and thus influences epigenetic regulation within the cell nucleus. The last thing we looked at for this preliminary experiment was histone H2B. We looked at the chromatin organizational structure changes within the nucleus using this histone for reference. So we quantified the euchromatin condensation density of this histone using super resolution imaging. And we use this to examine the changes in transcriptionally active chromatin organization within the nucleus after FSS. So with our STORM results, we didn't see any observed changes in the organizational structure of histone H2B. And this could be simply because we only ran the flow experiment for 15 minutes. So while this was enough to influence the histone modifications and their epigenetic properties, it may not have been enough to influence the chromatin organization within the nucleus. If we would have seen a change in chromatin organization, we would have seen the representative images reflect more of a red or orange or yellow condensation, but here we still see blue across all the parameters. So since we saw the most results in our preliminary experiments at high FSS, we were prompted to do a secondary experiment only utilizing high FSS. And in this secondary experiment, we treated the cells with an LPA treatment to explore the effects of cell contractility and GCPR-induced activity on histone modification regulation. I'm not sure why this made this last bullet fall down, but nonetheless, previous research findings have already concluded that LPA induces YAP translocation. So for this secondary experiment, we did not use YAPs. So here's a drug treatment workflow for the secondary experiment. It's the exact same as the primary experiment, except for on day 1, we treated the cells with LPA for 1 hour and incubated them and then ran the flow. And again, LPA is a drug that increases cell contractility, and we treated the cells with LPA at 50 micromolar for 1 hour. So after our LPA treatment, we found that across the board, increased cell contractility decreases the expression of both histones. However, for the trimethylated histone, we found an unexpected result. So as you guys can see here on our graph, at high FSS, the expression of the trimethylated histone was increased as compared to the LPA control, which is not something that we had seen in the untreated drug groups or even in the drug group with the acetylated histone. But that was unexpected because on any other flow experiment, typically high FSS made the expression of the histone lower than the control. So in conclusion, we were able to conclude that YAP nuclear localization highlights FSS's activation of GCPRs, since we saw the mechanosensitive properties of GCPRs lead to YAP regulation within the nucleus. We also found that mechanical stimuli is linked to epigenetic regulation. So since FSS regulated the histones through GCPRs, we were able to conclude that the epigenetic regulation was due to FSS, and we also observed this in the LPA treatment group. So even with induced GCPR activity and induced mechanical stimuli, the histones were still regulated through FSS. And finally, we concluded that FSS altered the expression of both histone markers, which means that FSS can lead to altered transcriptional regulation, which is observed in atherosclerosis, since that plaque that's blocking the blood vessels causes FSS. So future directions for this project. Since this was a preliminary experiment, we used a HeLa cell line, since that is the standard for studying mechanical transduction, but endothelial cells could be used in the future, since they are the cell type that lines blood vessels. A softer surface to better mimic blood vessels could also be used, since we use a glass slide and a microfluidic device that was also glass. So maybe something like a hydrogel, as Caroline discussed earlier in her project, could be used. Also, extended FSS duration could probably cause more global changes. Most of the changes that we observed were more local, but at a time that's longer than 15 minutes of flow, we could probably see more global changes. In addition to that, a longer flow could also probably allow us to see more changes in the chromatin organizational structure within the cellular nucleus. And we could also employ super resolution imaging to explore if altered cell contractility affects histone organization. So after the drug treatment, we didn't use STORM, but that could be a good way to see if induced cell contractility makes a difference as compared to regular FSS. And finally, ChIP-seq could be utilized to quantify gene regulation at specific loci. So again, since most of the changes that we've seen were global in this project, local in this project, ChIP-seq could see the specific genes that are being regulated. You guys can't see it because it's cut off on my screen and on here, but two of the genes of interest would probably be apolipoprotein and gene MIA3, because those genes have already been found to be associated with atherosclerosis and to be transcriptionally regulated in patients with atherosclerosis. And finally, I would like to acknowledge, of course, everyone in the HEO lab, especially my two mentors, Felicia and Tyler, and my PI, Dr. HEO. And of course, I would like to acknowledge the CEMB. Thank you guys for allowing us to be here this summer, and NSF. And then here are my references. I'm happy to take any questions.
Not quite sure what dyne is, DYNE.
Oh, a dyne is just a unit of force. That's just the unit of force that we use. Oh, yes. Any questions? Okay, thank you guys.
So our next speaker is going to be Piseth Coy, coming to us from Johnson and Wales University. This summer, Piseth was in the metal lab. And today, Piseth's going to tell us about 3D in vitro neuromuscular junction modeling.
Hi everyone, my name is Piseth and I'm in the metals lab. So my summer project is 3-dimensional in vitro neuromuscular junction modeling. So a little bit of background of neuromuscular junction. It's the specialized synapse that is responsible for muscle contraction. It's triggered by electrical signal called action potential that travel through the moneuron, causing the calcium gate channels to open, and upon entry of the calcium, it's caused synaptic vesicle containing the acetylcholine to release through the process of exocytosis and bind to the acetylcholine receptor on the sarcolemma of the neuromuscular of the skeletal muscle. So the pictures on the right is showing how beautiful the neuromuscular junction of a tissue. So as we know that our extracellular matrix is constantly undergo remodeling that's involving cross-linkage, breaking down and synthesis of the new ECM component. In the context of aging, the neuromuscular junction undergoes a process of degeneration and remodeling, as we can see that we have like decreased amount of Schwann cell that responsible for maintenance and regeneration of the tissue, and also a widening of the synaptic cleft over here that's disrupt increase the fragmentation and disrupt the acetylcholine receptor on this. So the most common cause of sarcopenia, which is due to natural aging, is involved in reduction of the muscle density and infiltration of the fat, as shows in this. This is a healthy individual, and this is an older individual. So although exercise has shown to slow down this process, better understanding of the mechanical property of the extracellular matrix and development and maintenance of the neuromuscular junction, we could better understand the context of aging and fibrosis. So what what do we propose? So we hypothesize that mechanical property of the extracellular matrix affects the neuromuscular junction development. So to tackle this hypothesis, we decided to design a three-dimensional in vitro modeling mimicking the native environment of the neuromuscular junction. And the reason we choose 3D in vitro modeling instead of conventional 2D is that they are consequent that's grown on the monolayer as the lack of extracellular matrix and cell interaction and the change in geometry and organization of the cell that can affect overall cellular development and behavior. And also 3D is like better mimic physiological relevant of the these are some of the previous 3D modeling using the PDMS mold and microfluidic device. So that's allow us for using custom material to study mechanobiology. So I'll go through the experimental flowchart. So first we induce the iPSC, which is induced pluripotent stem cell. It's a reprogrammed somatic cell. We are using different neurotrophic factor and differentiate it all the way to the mononeuron progenitor cell, and then seed it into the 96 plates and spin it down. And after 14 days, form a mature, and then we co-culture in the fibrin gel tract and load it into the PDMS mode. So this is the cell identity of each state before spheroid formation. We have on day 0, iPSC, we're looking for a defined border and prominent nuclei. As we go along, we don't see really much morphology changing, but on day 18 specifically, we saw a neural light projection. So in order to confirm identity of each state, we perform immunostaining. GAPI, which is staining for nuclei. SOX1 is the early marker for the neuroprogenitor cell. And so neuroprogenitor cell, we saw expression of GAPI and SOX1 and no expression of AMNX1 and OGLIC2, which is the transcription factor that expressed in the mononeuron downstream sulfate. And then we also stain mononeuron progenitor cell expression. We saw expression of DAPI, SOX1, and OGLIC2, but no expression of AMNX1 yet because it's not fully terminally differentiated. It's still in the progenitor stage. So this is the result for spheroid formation. On day 18, we see 5,000 cells in the and then centrifuged, and then this is 1 day after. It's formed a cluster called spheroids through the process of integrin protein and some bonding. And this is after 14 days of maturation. It's highly compacted. And then we perform another immunostaining to confirm expression of AMNX1 to like further do functional analysis and co-culture with the myoblasts. So a little bit about the hydrogel. So in order to 3D in vitro modeling, we incorporate into these fibrin gel tract network. It's the fibrin is the key protein that involved in blood clotting, and wound healing is the network is formed by conversion of the fibrinogen to the fibrin monomer through the enzyme thrombin, and then polymerize to form a fibrin scaffold. And this is we use this PDMS platform thanks to Serchen, one of my lab-mate in Adol's lab. So on this left compartment, we co-culture with mononeuron spheroid and human myoblasts in the gel tract. hydrogel, and this one is just pure myoblast. After 14 days of differentiation, we don't see any formation of the myotube, unfortunately, but I wish I have more time to get into it. And then, so our conclusion, our first objective is to design a three-dimensional in vitro modeling, mimicking the native environment of neuromuscular junction. What we able to accomplish is successful generation of the mononeuron spheroid, and we're still in the progress of testing for suitable platform to form the neuromuscular junction. And our limitation of the study is the degradability of the fibrin gel, which is typically 15 days, and we can exactly precise control over the mechanical property of the fibrin gel. And our future direction is to choose an ideal platform for formation of neuromuscular junction, such as organ on the chips or more PNMS mold fabrication. And also, we also want to do further optimization of the on-demand tunable hydrogel to get a on-demand stiffness and relaxation through, for example, photoprotective crosslinker chemistry. Yeah, so takeaways message, we believe that our research has the impacts to three-dimensional disease modeling, drug screening application, and therapeutic transplantation, but it's required more advance in bioengineered materials. So I would like to acknowledge CMB for making this program possible for us, and Metal Biomaterials Lab for the opportunity to me to work in the lab, and especially my PI and Albert for having me in his part of his project. So thank you. Any question? Yes. Amanda.
I wanted to study purpose was the investigation different mechanical properties on this differentiation pathway, this differentiation.
Yeah, so first of all, we use the fibrin gel track hydrogel as like we try to make a control first, and then we also test for like a suitable platform, and then until we successfully form the myotube and the neuromuscular junction in that gel, we're gonna go over to the tunable hydrogel one using the photo cross-linkage chemistry, which
You mean you try different different fibrin gel with different mechanical properties?
yeah, but the fibrin gel is not, we can't really like precisely control over the so that's why in this we just in the testing stage and try to have like a control group before we turn over to the tunable mechanical property, and then we do some functional assays like quantification of the acetylcholine receptor and all of that, and then be able to like model some more neuron degeneration disease, all of that. Does that answer your question? Okay.
You may have answered the question a little bit already, but I was just gonna ask, like, this model at the neuromuscular junction.
Yeah.
What types of diseases?
So one of the study that I read about is they use they do the ALS disease, which is amyotrophic lateral sclerosis. It's a motor neuron disease, and by successfully developing a 3D in vitro modeling, we could eventually go over to the patient-derived and then model ALS disease and study the effects of the gene expression and all of that.
Our next speaker is going to be Yariani Dennis Vasquez from the University of Puerto Rico, Calle. And this summer, Yariani was in the Disher lab, and today is going to tell us about investigating the role of P10 and myosin 2A in regulating genomic instability in lung adenocarcinoma and melanoma cells.
Hello, everyone. My name is Yara Veres. I'm from the University of Puerto Rico, Calle, and this summer I had the pleasure of working in Dr. Disher lab along the mentorship of Alicia Enlas. And I will be discussing my project investigating the role of P10 and myosin 2A in regulation of genomic instability in lung adenocarcinoma and melanoma cells. So cancer progression is driven by hallmarks of cancer, which one of those are avoiding immune destruction, enabling replicative immortality, and resisting cell death. And we were most interested in genomic instability, which is caused by genetic mutations, chromosomal rearrangements, cell division errors, and aneuploidy, which is an abnormal number of chromosomes in a cell. And all of this drives tumor development and malignancy. Across different tumors, there's a variability of genomic instability and aneuploidy. As you can see, breast cancer, for example, has a higher level of aneuploidy. And there are some other types of cancer that can have a lower level of aneuploidy and be caused by smaller genetic level insertions or deletions. One of those cell lines that we worked on was A549 cell line, which is the lung adenocarcinoma. And this one is known by low genetic homogeneity and high aneuploidy. And it also has a pretty rapid doubling time of 22 hours. The other cell line that we worked on was the B16 cell line, which is mouse melanoma. This one is known to be highly metastatic, exhibits aneuploidy, and has high chromosome instability. One of the ways genomic instability can manifest is through micronuclei formation, which is when chromosomes missegregate during cell division and small extranuclear bodies separate from the daughter nuclei. And we focus on perturbing tumor suppressors with drug treatments to inhibit their function to assess the effect on genomic instability. We use 4 different drug treatments. These were reversing, which inhibits the spindle assembly checkpoint, BOPIC, which is the P10 inhibitor, which induces mitotic arrest, devastatin, which inhibits non-muscle myosin 2A by disrupting actomyosin interactions, and palbociclib, which is a CDK4 and CDK6 inhibitor, which blocks cell cycle progression from G1 to the S phase in the cell cycle, and is actually used for breast cancer treatments. So the mechanisms of tumor suppressors of myosin 2A is by binding to the actin filament and promoting cell contractibility. So by inhibiting this myosin 2A with blebistatin, we will see membrane bleibing and apoptosis. And as for the P10 tumor suppressor, it acts as a tumor suppressor by inhibiting the PI3K and AKT pathway and inhibits the apoptosis inhibition. Our experimental setup was using A549 cells and B16 cells, culturing them in T25 flags and passaging them into 6-fold plates. In total, we had 3 6-fold plates. The first one had reversing, BOPIC, blebistatin, and the control treatment, which was DMSO, and this was left for a 24-hour treatment. The second one was with CDK4/6 inhibitor and DMSO, and this was left for a 48-hour treatment. And for the B16 cells, they were treated with reversing and DMSO for a 24-hour treatment. Then we proceeded to immunostain with DAPI to view the DNA and following to view the F-actin. Then we proceeded to do fluorescence microscopy and view the cells in 9 frames to quantify micronuclei and mitotic cells. The results present that reversing suppresses mitosis but increases micronuclei in A549 cells. As you can see in the graph, there's an increase of 15% of micronuclei while suppressing mitosis to a 1%. The green arrows point to mitotic cells in the late stages of mitosis, and the yellow arrows point at micronuclei. As for the CDK4 and 6 inhibitor treatment, it inhibited mitosis and increased micronuclei. And for the reversing treatment on the B16 cells, also suppressed mitosis and increased micronuclei. Another side of our project, we initially wanted to engineer chromosome reporters by tagging genes with GFP to evaluate chromosome loss. And we wanted to insert GFP into these select genes to establish the chromosome loss. And those genes were lamin B1, which is on the chromosome 5 and expressed at the nuclear lamina, paxillin, which is on chromosome 19 and on the focal adhesions, and fibrillary, which is on chromosome 12 and expressed at the nucleolus. Some of the steps that we took were culturing GFP positive E. coli and inoculating them, incubating them, and then we centrifuged and we suspended the cells, lysed the cells, and neutralized them. Then we put it on a spin column to obtain the plasmid DNA. Then we measured the concentration in a nanodrop spectrophotometer. And our conclusions are that reversing treated A549 cells and B16 confirm the importance of the spindle assembly checkpoint, which when inhibiting, it's evidence that micronuclei forms and mitotic is suppressed. And CDK4/6 inhibitor effectively halts cell proliferation in A549 cells, which suggests that the tumor progression is slow, and nonetheless, the presence of micronuclei indicates the persistence of genomic instability without ongoing cell division. Finally, the inhibition of PTEN and myosin 2A may not directly induce genomic instability, though their influence on other cellular processes remains a consideration. And for future directions, for this study, we include incorporating other approaches to induce genomic instability, such by squashing cells or altering the stiffness of the microenvironment, and also continuing the CRISPR-Cas9 gene editing technology to knock out genes to further investigate their specific roles. And special thanks to the Center for Engineering Microbiology and the Digital Lab for their help and support, especially my mentor Alicia and Dr. Dennis Fisher. Thank you. Any questions?
So our next speaker is going to be Kehinde Alowepo from Alabama A&M University. And this summer, Kehinde was in the Berkeley lab. And today, Kehinde is going to tell us about optogenetic tools for studying 3T3 and HeLa cell mechanobiology.
So I'll be giving a talk on optogenetic tools for studying 3T3 and HeLa cell mechanobiology. So why is the active cytoskeleton even important? Active filaments are crucial for the movement of cells because they enable processes like migration. And this process helps cells move to one site and aids in tissue repair. So by using light to control wave signaling, we can precisely regulate actin dynamics to study cellular behaviors. The active cytoskeleton is also essential in mechanobiology providing structural support, enabling cell movements, generating and transmitting force, and allowing cells to sense and respond to mechanical signals from the environment. To delve deeper, we need to understand what ROA is. ROA is a small GTPase that when activated, it plays a crucial role in regulating the actin cytoskeleton. Once exposed to light, ROA moves to the plasma membrane. This movement is essential for its interaction with downstream targets. And the plasma membrane, ROA stimulates the formation of actin filaments, leading to actin polymerization, and this causes the cell to change its morphology and contract. These changes in the actin cytoskeleton can lead to the activation of YAP, which is a key transcription factor. Activated YAP moves into the nucleus where it influences gene expression related to growth. Now let's see how we can use optogenetics to precisely control reactivation and study its effects. Optogenetics is the use of light to control cells that have been genetically modified to express light-sensitive proteins. We're using optogenetics because we can precisely control specific signaling pathways at the subcellular level and temporal position down to the second. To understand this better, let's look at how one specific optogenetic tool, Bisolol-4, works in detail. So Bisolol-4 is a protein produced by the fungus Botrytis cinerea acinia affecting the fruits. In a variety of cell types, Bisolol-4 can move from the cytosol to the plasma membrane in response to blue light. So on the left of this image, we see Bisolol-4 in a dark adapted state where it remains in the cytosol. On the right, exposure to blue light causes Bisolol-4 to translocate to the membrane. So this reversible and light responsive behavior of Bisolol-4, which is a great tool for controlling protein locations within cells. So next, we'll see how we can use the property of Bisolol-4 to control the recruitment of specific proteins such as ROA to the membrane. So here we use Bisolol-4 to control the location of ROA, which is a critical regulator of the actin cytoskeleton. So in the dark adapted state, our protein of interest is located in the cytosol and it remains inactive. But upon blue light exposure, Bisolol-4 moves to the membrane and brings the protein of interest with it, resulting in localized activation. So this diagram illustrates the process. So on the left, you see the protein of interest is inactive in the dark, and on the right, you see blue light triggers the movement of Bisolol-4 and the protein of interest to the membrane. So using Bisolol-4, we can precisely control reactivation in cells, as demonstrated in our previous studies with HEX cells. So here you can see the HEX cells in the dark adapted state. So on the left and on the right, you can see after blue light stimulation, ROA is activated, leading to changes in the cell morphology. So this image shows the clear transition and provides evidence that light can effectively control reactivating cells. So in the right image, you see like the cells contracting and there being like this junction that's forming. So that's what we expect to see. So building on this result, we're extending our research to HeLa and 3T3 cells to further investigate optogenetic control of ROA. So to achieve this, we use the process called transient transfection. We have two plasmid constructs, one expressing the optogenetic protein OptoROA and another expressing Bisolol-4, which serves as the negative control. And both plasmids were repeated in E. coli and they're purified to ensure high quality DNA for transfection. So we transmitted the HeLa and 3T cells with the purified plasmid so we can observe the expression of the optogenetic proteins. Post-transfection, we use the fluorescence microscope to stimulate the cells with a specific wavelength of light and monitor the expression of the target proteins in the cell to confirm successful transfection. And here you can see how we capture these images and analyze the cell contractility. Now let's observe the video showing the cellular responses to optogenesis activation in both HeLa and 3T cells. So we use Bisolol-4 as the negative control. This means it doesn't activate the ROA signaling pathway, which helps us compare and understand what happens when the pathway is not triggered. We don't expect Bisolol-4 to cause any noticeable changes in the cells when stimulated with light. And on the other hand, with OptoROA, we expect to see changes in how the cells behave, like the cell contracting and changing in shape when stimulated with light. So then this graph shows how much the cell area changes. So for Bisolol, as you saw in the video, there wasn't much of a change as expected because it is the control. And this confirms that Bisolol is a good control. And then for the OptoROA, we also saw that there was a big change, which is not what we expected. But we can also conclude that optogenetic tool did not work best in our HeLa cells. So this could be due to cytoskeletal dynamics and could also highlight potential challenges with the plasmid constructs that we used. So we did the same experiment in 3T3 cells. Again, Bisolol-4 is our negative control, so we don't expect to see any changes. And then in the OptoROA, you can observe, and then the OptoROA, we did the same experiment in the OptoROA. And then you can see, we can observe the cell contracting and like changing in shape. And this is what we expect to see. We expect to see the cell changing the morphology. And this graph shows a significant change in the cell area for the OptoROA group from before it was exposed to light and then 10 minutes after the cells were stimulated with light. So this shows successful OptoROA activation because the cells were able to like change their morphology. So we also conducted an experiment showcasing the intensity of phalogen staining. So phalogen staining is a technique used to label filamentous actin, F-actin in cells, allowing for the visualization of the actin cytoskeleton. So this diagram shows the pathway where ROA activation leads to actin polymerization. So this data shows that the active elements have been stayed with the following. This data shows the active elements that have been stayed with following. On the left, we see that the 2 cells expressing up to rate in the dark states with lower following intensity, indicating less active polymerization. And then the arrows you see are pointing to the areas where they're labeled M cherry. So these are the active elements that are visible. So this is showing the baseline of the active polymerization. On the right, after 2 hours of blue light illumination, there's a significant increase in the following intensity. So this is showing the enhanced active polymerization. So the arrow again is highlighting the regions where there's an increased fluorescent signaling, indicating higher collagen binding and then indicating an increased active polymerization. And so this increase in fluorescent intensity indicates changes in the active element's organization and density upon rare activation. So YAP is let's talk about YAP now. So YAP is a transcriptional coactivator which regulates growth. So the diagram here shows how rare activation leads to YAP moving into the nucleus where it can regulate gene expression. So YAP's nuclear localization is a critical step in its role as a transcription factor. So when YAP is not active, it is located in the cytoplasm, unable to influence gene expression. However, factors like the cell contracting due to off-to-rare activation can cause it to translocate to the YAP. So can cause the YAP to translate from the cytoplasm to the nucleus and cause it to promote the expression of genes involved in cell growth and proliferation. So this can also be helpful in processes like wound healing where we need increased tissue growth. So in these images, again, we have the 3T3 cells expressing up to a fixed staying with a YAP antibody. In the dark state, which is the left image, YAP is mostly cytoplasmic, and the arrows there are indicating where YAP is visible in the cytoplasm. So after 2 hours of blue light stimulation and activation of OA, the arrows indicate that YAP has translocated to the nucleus, as shown by the increased nucleus staining in the right image. So YAP goes from the cytoplasm to the nucleus, and that's how you know that okay, YAP is active. So to conclude, we successfully implemented an optogenetic tool to control reactivation in 3T3 cells, and our experiments demonstrated that blue light stimulation significantly increases active polymerization as indicated by the phallogen staining. Additionally, we observed the translocation of YAP from the cytoplasm to the nucleus, confirming the activation of YAP signaling in response to reactivation. But for future directions, we could possibly do a wood-in assay to further explore the impact of the functional impact of reactivation. So by using our optogenic system, we can precisely control reactivation and investigate how enhanced active polymerization influences the wound healing process. Another promising future direction is to delve deeper into YAP mechanical signaling. So given that YAP translocates to the nucleus upon reactivation, we plan to investigate how these mechanical cues in the cellular environment can also affect this YAP activity. So finally, I would like to acknowledge the contributions of the Berkeley Lab and thank them for all their support, as well as the CMB team for a great summer of research. Thank you for your attention, and at this time, I'll be able to take any questions you might have. Yeah.
So the cells changing morphology, what was the time scale of these videos?
It was 10 minutes. So it was like post, we took it from T0 to 10 minutes. It's also like as in the cell area. So that's when you saw the graphs. That was the difference in cell area from the T0 to 10 minutes. Yeah.
Did you have to make the plasmid that you put in the light?
Yeah. Yes, because they're those are our BCO4 and then our auto-way tools. So they have to be in the plasmid so we can transfer the HeLa and 3T3 cells with it.
So you built that first.
Yeah, my mentor. Yeah, my mentor. Yes. Okay.
So our last speaker is going to be Denise Martinez from Drexel University. This summer, Denise was in the mock lab, and today he's going to tell us about mechanosensitive pathway activation following intervertebral disc injury.
Well, my name is. My name is Denise Martinez. This summer I have been doing an RE through CMB and Buck Labs with Kevin and Emily as my mentor. During this time, I have been investigating mechanosensitive pathway activation post-invertible disc injury. The invertible disc supports spine load and flexibility, which is essential for everyday mobility. Lower back pain is the leading cause of disability globally, so this research is very impactful. Mechanical stimuli from spinal motion can lead to degenerative changes in IBD. IBD herniations where the nucleus pulpus and P extrudes beyond the annulus fibrosus AF results in significant cell death and tissue remodeling. Understanding the cellular processes and this herniation is crucial for developing treatments. Mechanosensing of IBD cells within injured or diseased environments trigger extracellular matrix degradation and inflammation. So during our investigation, we focused on mechanoactivation in vivo models of AF injury and identifying key mechanosensing mediators. We hypothesized that mechanosensing pathways, YAC, TAZ, RO, and ROC would be increasingly activated in injured IBDs compared to healthy IBDs. We began this process by mapping out a plan of action, focusing on two groups of mice, group 1 being the injured and group 2, the control being the baseline. Our study has the potential for multiple key endpoints, but during this time, or during this presentation, I will only be focusing on endpoint 1 at the 2-week mark. At the endpoint, we assessed two primary outcomes: inflammation measured through histopathology and gene expression analyzed using PCR. Specifically, we will be looking at mechanosensitive pathways and their role in disc degeneration. The diagram below illustrates our study design. We have categorized the disc into surgically injured, adjacent, and baseline, providing a comprehensive view of how injury affects both the directly injured and adjacent disc over time. This design helps us understand not only the localized impact of injury, but also how the altered mechanics of the spine influences adjacent discs, potentially leading to further inflammation. By following this plan, we aim to understand the mechanobiological responses to AF injury and identify potential therapeutic targets to mitigate disc degeneration and lower back pain. Our experiment design involves a detailed approach to studying the effects of AF injury on IBDs. As illustrated in panel A, we focus on two specific injury sites within the spine, disc 5-6 and disc 7-8, along with their adjacent discs. Panel B illustrates how we were able to accurately perform the surgery through the use of X-ray guidance. Injured discs and their adjacent counterparts are isolated 2 weeks post-surgical injury. The discs above the dotted line are analyzed histologically by staining sections with SAFO fast green, while the discs below the line undergo PCR to measure gene expression of 4 mechanosensitive pathways and 3 immune cell markers. This dual approach allowed us to comprehensively assess both structural and molecular changes post-injury. We're first going to take a look at the histological results of the study. The images on the left show histological sections stained with SAFO fast green to highlight cartilage matrix. Starting with the baseline IBD, we see a well-preserved structure. The AF exhibits distinct and organized concentric layers, indicating a healthy state. The NP is centrally located and it appears to be well hydrated, which is essential for normal disc function. In the adjacent disc, the multi-layered structure is largely preserved, but shows slight disorganization and disruption compared to the baseline. The NP remains intact, but appears slightly condensed. In contrast, the injured IBD shows significant structural disruption. The AF layers are disorganized with visible tears and separations. The NP is severely depleted and less distinct, indicating substantial degradation and loss of hydration. These changes illustrate the extent of degeneration following injury. The bar graph on the right quantifies the disc height across the different groups.
We observed that this height reduction is a notable feature following injury. The baseline discs have the greatest height, followed by the adjacent and then the injured disc. Importantly, while the reduction in disc height is significant between the baseline and both the adjacent and injured disc, there is no significant difference in disc height between the adjacent and injured groups. This suggests that while injury impacts disc height, adjacent discs also experience structural changes likely due to altered spinal mechanics. It would be interesting to see if this remains true if we look at different time points. These are gene expression results focusing on mechanosensitive pathways. The bar graph shows the fold change in expression levels of Rho, ROC, YAP, and TAZ relative to baseline discs. Starting with Rho, we observed a significant upregulation in injured discs compared to baseline and adjacent discs, indicating an increase in mechanosensitive pathway activation following injury. For ROC, there is a notable increase in expression in injured discs. The significant upregulation in injured groups compared to baseline suggests that ROC plays a key role in cellular response to AF injury. And then looking at YAP, we also see an increased expression in injured discs compared to baseline, although the difference isn't statistically significant. The trend supports the involvement of YAP in mechanotransduction responses to disc injury. TAZ expression is significantly upregulated in injured discs compared to both baseline and adjacent discs. The marked increase in TAZ expression highlights its role in responding to mechanical stress and injury within the IBD. Overall, in the mechanosensitive pathway markers, there was a significant upregulation in injured discs compared to baseline and adjacent discs. These pathways are crucial in cellular response to mechanostimuli, influencing processes such as cell proliferation, differentiation, and survival. The increased expression indicates activation of these pathways in response to the surgically induced AF injury. These are the PCR results for the immune cell markers we use to examine the inflammatory responses and immune cell infiltration following AF injury, as shown in the current slide. The bar graph illustrates the fold change in expression levels of the inflammatory markers, Ly6C, CD68, and F480 relative to baseline discs. We see a significant upregulation in injured discs compared to baseline and adjacent discs in all three immune cell markers, indicating an increase in monocytes infiltration and inflammation in response to injury. The significant increase in gene expression in the injured discs correspond with histological evidence of severe tissue disruption. This suggests that the macrophages that you can see there are playing a critical role in response to injury, possibly contributing to both the degradation process and subsequent repair mechanisms. Our study highlights key findings regarding mechanosensitive pathways and IBD injury. Mechanosensitive pathways such as YAP and TAZ and Rho and ROC have emerged as potential therapeutic targets. We observed significant upregulation of these pathways in injured IBDs, indicating their crucial role in cellular response to AF injury. This height reduction was noted post-injury, emphasizing the structural impact of AF injury. Our findings align with existing literature on extracellular matrix degradation and inflammation, reinforcing the importance of these pathways in this degeneration. In our future work, we will, if we have the opportunity, we would include multiple time points at 2, 4, and 8 week marks to better understand the temporal dynamics post-injury. We would study wild-type injured mice as well as CD14 knockout mice to investigate the role of CD14 in inflammation and disc degeneration. We would also use PA gels with varying stiffness, glass, 5 kPa and 55 kPa in an in vitro model to mimic changes in microenvironment post-injury. This approach would help us better understand how mechanical properties influence cellular responses. These studies aim to deepen our understanding of disc degeneration and inflammation and identify potential therapeutic targets. Thank you to Emily and Kevin for being such great mentors this summer. And thank you to everyone who organized this program and Muck Labs for being so welcoming.
Really nice presentation. So right now you presented the short-term results really nicely. With the longer term, do you expect there's to be more differences between the adjacent and the injured discs, or maybe do you expect like one to worse in the long term?
Honestly, I would say that the injured would be worse, but at the same time, like that might not be true because the difference in mechanics that occur because of the injury changes the way things are distributed between the loads are distributed. So it's possible that it could be worse or it could be the same or it could not be as bad, but yeah. It would be interesting to see.
Okay, well, first, thanks so much to all of the presenters, wonderful job. And thank you all for coming in person, and for those who are on Zoom, thank you for attending virtually. So that's the end of our presentations. Great job, everyone. We have a celebration luncheon outside, and for all of our wonderful UExB researchers, I have a small parting gift for you, and for all of our mentors, a small parting gift for you as well. So let's eat.